Hello, my name is Wyatt Barnes, and I am a student at BYU Idaho taking Bio 461. This is my first exam essay, and this video will be split into two parts. First, we'll be talking about homeostasis and how our body keeps us in prime condition despite changes in our external environment. And second, we'll be talking about transversing the cell membrane and how there are different ways to do that and how there are different proteins that are involved with that. So let's jump right into it. So part one, homeostasis. What is it? Well, homeostasis is basically our body maintaining an optimal internal environment that is relatively stable despite changes that happen outside or on the external environment. So one good example of this would be living in Rexburg. It gets really, really cold here in the winter, and despite it being really cold outside of our bodies tend to do a really good job of keeping our internal temperature relatively stable, usually around 98 degrees Fahrenheit. How does this happen? Well, we go through something called a reflex control pathway. The general idea of this is we have a receptor that senses changes in the external environment. The receptor is usually a cell of some sort of nervous tissue that senses a change in the environment. This change could be any number of variables, including temperature, pH levels, and blood pressure, etc. There could be a number of variables involved with this. When our receptor senses a change in the environment, say temperature, then it sends a message to what is called a control center. I'm going to draw a really bad picture of a brain here, but that's a brain. So a receptor senses a change in the environment and sends a message to the control center. Usually the control center is the brain. Now, this pathway is called an afferent pathway. A, with A as in apple, sending a message from the part of the body that is receiving information about the environment changing to the control center, which makes decisions based off of the information received. Next, our control center sends information to what is called an effector. Effectors are basically the parts of our body that do the control center's bidding. So say we're really cold. Well, our brain will receive information from some receptor towards the surface of our body that senses the change in our external environment getting colder. That information is then sent to an effector, such as our muscles, which begin to shiver to try and increase or maintain our internal body temperature despite changes outside of our body. That pathway is called efferent. E as an example. This is what we call a negative feedback loop. Negative meaning we are taking a change in our body and trying to bring it back to what is considered normal, or in other words, a set point. Our set point temperature is usually around 98 degrees Fahrenheit. When our external environment gets colder, our body temperature is dropping just as slightly before this pathway happens. When this pathway happens and our effectors begin to work, our temperature rises back to the normal set point. That is one example of a negative feedback loop. Now say this doesn't work. What if it's so cold that our body starts to shut down? Now that is when we get into really big trouble. That's when we run into what is called a positive feedback loop. Usually positive feedback loops are a really bad thing, and oftentimes they lead to extreme cases where a patient is in the process of dying or does die. How this can work is, say you lost a limb and you're losing a lot of blood. In order to make up for this drop in blood pressure, our body does a couple of things. Our heart rate rises and our blood vessels constrict. And we have an adrenaline rush to help with all of that. So how this works is our receptors will sense a drop in blood volume after we have a loss of a huge amount of blood. Then we go through the afferent pathway, and that information is sent to the control center. The control center will then send that information out, a command, through an efferent pathway to the effector, say the heart. The heart receives that information and increases in its heart rate in pumping. Now, if the hemorrhage is too much, the more the heart is pumping, the more blood is leaving the body. And hence, this loop becomes a very bad thing and can actually lead to death, despite the original plan of increasing blood pressure again. So, there are a couple of very few instances in which positive feedback loops are a good thing. One example would be during childbirth. During childbirth, the child pushes on the birthing canal, on the cervix, and that releases oxytocin. There are receptors on the cervix that sense that pressure from the baby as the contractions are happening, and it sends that up to the control center, the brain. Then the brain sends down information, oxytocin, to the effector, which is the muscles around the birthing canal and uterus, and that causes larger, bigger contractions, and it is a positive feedback loop as it is getting farther and farther away from our original set point. In this case, that's a good thing because we want our baby to be birthed. We want that baby to leave the mother's body. Now that we've covered part 1, let's cover part 2 of our video, which involves how things get across our cell membranes. One important thing to note is that our cell membrane, the membranes that make up all of our cells, are made up of a phospholipid bilayer. Here, I'm going to draw a very simplified version of what I mean by that. So, a phospholipid has phosphate heads. These are hydrophilic, meaning they like water. They like to stick to water, and they don't like things like fats and lipids. But they have these little lipid tails facing on the inside of the membrane. And these tails, which are made up of lipids, are hydrophobic. Hydrophobic meaning they fear water. They don't like to be around water, they repel water, they push it away. So anything that is similar to water, meaning anything that is charged, anything that is particularly large, anything that is polarized, will not be able to go across our phospholipid bilayer very easily. Now, this could be the extracellular space. Let's see, I drew that backwards. Extracellular space. And then this could be intracellular space on the bottom. Now, it is a bilayer, meaning there's 2 layers to our phospholipid bilayer of our cell membrane. So each layer has the phospho part, the phosphates, or the water-loving part, facing on the outside, and the lipid layer part facing inwards, like so. Here we have the extracellular layer and the intracellular layer. Notice how the phosphates heads are on the outside of both layers, and the tails, which dislike water, it pushes it away, are on the inside. So say we have a sodium particle on the outside of the cell. Our little sodium particle has a charge. Now that means it cannot easily go across our phospholipid bilayer. Luckily for our sodium, we have what is called a sodium channel. This is one example of the many different types of channel proteins that exist within our cell membranes. Now, for channel proteins, they're often extremely specific, meaning they only let across one or two things through a specific channel. So this is a sodium-specific channel protein. I didn't draw it very well, but this is supposed to illustrate a tunnel, if you will, for the sodium to traverse our phospholipid bilayer membrane. Many channels are voltage gated, and they have gates. So say this channel is voltage gated, it'll have some sort of gate, let's say a gate down here that kind of looks like a ball and chain. Once it senses a change in the charge surrounding that gate, a very specific charge that is specific to a sodium ion, it will open up and allow that ion to either leave or enter through that channel. Now, we also have something that is called a ligand-gated channel. Ligand-gated channels act very similar to how voltage-gated channels act, except they have a little keyhole, if you will. Say that's our keyhole. And we have a gate covering the outside of our channel. And we have a ligand that is the perfect shape, or the perfect key, to fit into the site there. Once that ligand locks into place, it opens up the gate and it allows the specific solute to go through that specific channel. The last type of channel protein that we have is something called a mechanical gated system. Mechanical gated channels act similar to the other two types of channels, except instead of using a ligand or a charge, it responds to pressure or temperature or something of that nature. This is how we're able to sense touching things. We can sense textures, different types of textures, because of this type of channel protein. The pressure that we receive on our fingertip cells opens up a gate that is able to allow passage for communication to happen. And then that communications run through our nervous system up to our brain, and we're able to perceive what we know as different textures. Now, this is what we call facilitated diffusion, meaning we're moving from a concentration that is higher to a concentration that is lower. It doesn't actually require any energy. Cell energy is usually in the form of what is known as adenine triphosphate, or ATP. ATP is only used in the situation where we have solutes going from a low concentration, pushing them up into a high concentration. Now, why would we do that? It seems like it wouldn't be very energetically favorable to do that. But there's a trick that we like to do. A really common example of this would be the sodium potassium ATPase pump. And how this works is we have a channel that pushes sodium out of the cell. So say this is our pump right here. The two blue lines are our phospholipid bilayer of the cell membrane. And say we're pushing 3 sodiums, the orange dots can be sodiums, out of the cell. And for every 3 sodiums pushed out of the cell, we have 2 to potassium particles entering the cell. And over time, we get a lot of sodium ions sitting outside of the cell. Now this would require Now this process requires energy in the form of ATP. ATP is used up to create a high concentration of sodium particles outside of the cell and a low concentration of sodium inside of the cell. This creates what is called a chemical and charged gradient. Sodium ions each have a positive charge on them. This positive charge is really building up outside of the cell, and it really wants to be let back into the cell. But because of the specificity of our channel protein, it won't allow that sodium to go back into the cell. And so we're, energy speaking, we're going uphill. And it's creating a big concentration of sodium outside. Now, why would we want to do something that is not energetically favorable? Well, the answer lies in what happens next. Somewhere down the line in our phospholipid bilayer, we have another protein that is going to use that concentration of sodium outside of the cell, similar to how we use turbines to collect energy from wind. So those particles are really going to want to be let through, and they're going to go through and run really quickly through this channel back into the cell membrane. As this happens, this protein is able to convert that energy, that charge, as it goes through, to create more ATP, or more energy. And it actually becomes extremely energetically favorable. Now, even though there is no energy used directly in the process of letting this sodium back into the cell, because of the concentration that was built up here, we still consider it an active transport, or something that's called secondary active transport. This would be a primary active transport where we are primarily using up energy to build up a concentration and a chemical gradient, and then this would be considered secondary active transport, where we are not using any energy directly within that process. Now, since we've gone through all of that, and we've talked about what it means to go across the cell membrane, how that happens, and the different channels that are used with that, let's talk about osmosis, which is basically the movement of water from a high concentration to a lower concentration. Now, this might seem like a really simple concept, but it can actually get a little tricky once we add some factors into here. So say I have a big tub of water, and within that tub of water, let's say I have a semi-permeable membrane. That's the orange line. Semi-permeable meaning water is allowed through that membrane, but nothing else is. And let's say we have a couple of chlorine ions in both sides of the membrane. Now, if we have equal amounts of chloride ions on either side of the membrane, then nothing happens. The water is going to stay the same. But say we have very few chloride ions on one side of the membrane, and a lot of chloride ions on the other side of the membrane. There's a really high concentration on this side and a very low concentration on this side. Then what's going to happen? Well, what will happen is water will move towards the higher concentration to try and dilute it to reach an equilibrium where we have a similar concentration on both sides of the membrane again. So, we'll have water leaving the side with very few chlorine ions, and a lot of water will go on the side with a lot of ions. In essence, water follows the higher concentration in an effort to make an equilibrium where both sides are in the same concentration levels. This happens within our cells all of the time. And because sometimes solutes such as chlorine ions or sodium ions or potassium ions are unable to pass through our cell membranes, water makes up the difference to try and create an equal concentration on both inside of the cell as well as outside of the cell. Now, what that means is when we have a cell that has a lot of sodium in it, and there's not a lot of sodium outside of the cell, in the extracellular space, say there's like very few compared to inside, then water will move from outside of the cell into the cell to try and even out the concentration. When this happens, the cell will be stretched and its volume will increase. It will become much larger than normal. This is what we call hypotonic. Now, tonicity refers to the cell's response after osmosis has already happened. To say a cell is hypotonic means it has swollen, or its membrane is like very large and to the max. Now, normally we want our cells to not be like that. Otherwise, we run the risk of having them pop or lyse. When we have a cell that is normal, that's something that is called isotonic. Iso meaning the same, and tonic just meaning the tonicity of our cell membrane. Isotonic is kind of what we want all the time. If we're in isotonic zone, we're in the safe zone. Now let's say we have a lot of sodium outside of the cell and very few sodium particles within our cell. Which way is the water going to go? Well, if we follow our pattern, the water will try and dilute the extracellular space and even out the concentration, so we'll have water leave our cell. This will create what is called crenation, or a hypertonic cell. Notice how our cell looks more like a shriveled raisin. This is what happens when water leaves the cell. We lose volume and it becomes hypertonic, and it looks kind of like a crinkled raisin. This can be extremely dangerous as it dehydrates our cells and can lead to death in extreme cases. Thank you for watching my video, and hope to see you next time.